Hi, my name is Eliana Longo, and today I'll be talking about alcohol and cancer risk. The purpose of my research was to determine if consumption of alcohol causes an increase in cancer risk. You might ask yourself, what is cancer? Well, according to the National Cancer Institute, cancer is a disease where some of the body's cells grow uncontrollably and spread to other parts of the body. Cancer can occur due to genetic changes in the body, such as cell division errors, genes inherited from parents, or damage to DNA caused by harmful substances. What is alcohol? Many people know what alcohol is, and many people consume alcohol. Alcohol is a very normal thing to socially consume as an adult, and many young people illegally consume alcohol and abuse it at the same time. The World Health Organization states that alcohol is a toxic, psychoactive, and dependence-producing substance and has been classified as a Group 1 carcinogen. Now you might be wondering, how does alcohol consumption and cancer connect? Drinking alcohol is such a casual thing, but it dramatically increases the risk of certain cancers, such as mouth and throat, larynx, esophagus, colon, rectum, liver, and breast cancer, according to the CDC. Going back to my research question, does consumption of alcohol increase cancer risk? I had to look at different experiments to determine whether or not research shows that increased alcohol consumption shows an increase of cancer risk. In the first experiment that I looked at, the goal of the study was to determine if the consumption of alcohol by postmenopausal women elevates serum levels of hormones that are associated with an increased risk of breast cancer, and it was a controlled feeding study. In Experiment 1, researchers had participants that were 51 postmenopausal women who were not using hormone replacement therapy rotate through 3 8-week dietary periods. During the 3 8-week rotations, the participants were randomly assigned to either consume 15 grams of alcohol per day, 30 grams of alcohol per day, or consume an alcohol-free placebo beverage. Then the concentrations of estrone sulfate and DHEAS were measured to conclude results found in the study. The results found were that when women in the study consumed 15 grams or 30 grams of alcohol per day, the estrone sulfate concentrations increased, as well as the DHEAS concentrations. The results conclude that postmenopausal women could increase their risk of breast cancer if they are consuming 1 to 2 alcoholic beverages per day, which shows that alcohol consumption increases risk of cancer. In the second experiment that I looked at, the goal of the study was to see if there's a connection between consumption of alcohol and changes in metabolic pathways, and when they are altered, if it increases the risk of developing breast cancer. This experiment is a human, animal, and cell study. The methods used to draw a conclusion of whether or not alcohol intake changes metabolic pathways, overall increasing the risk of cancer, used a method of administering carcinogens to some and not to others. The results concluded that ethanol converts acetaldehyde in mammary tissues, which has an effect on tumor development. It suggests that ethanol and acetaldehyde may alter the metabolic pathways, which increase risk of developing cancer, as well as an increase in hormonal receptor levels and cell proliferation, which can increase cancer risk. This study is linked with my research question because it found that ethanol converts to acetaldehyde, which is linked to tumor growth. This indicates that alcohol can lead to an increased risk of cancer development. In my final experiment, the goal of the study was to identify microRNAs that are dysregulated in alcohol associated with HNSCC, which is head and neck squamous cell carcinoma, and to investigate their contribution to the malignant phenotype. This was a human cell study. The method used in this experiment was 136 HNSCC patients' RNA sequencing data and compared to the expression levels of 1,046 microRNAs between drinking and non-drinking cohorts. The results found that alcohol induces the abnormal regulation of target genes involved in cancer invasion, which might play a role in head and neck squamous cell carcinoma pathogenesis and progression. This experiment shows how alcohol can aid in the development of tumor cells. To conclude, my research shows that increased alcohol consumption can lead to an increased risk of developing cancer. Thank you for watching.